Good. Okay, so welcome. This is week 12, and today we are going to review chapter 14. For me, this is the most significant chapter for your education. If you want to pursue nursing or a career that requires you to be vigilant about infectious diseases, okay? For me, this is the core of this microbiology class. After this chapter, we will see the immune system, we will see vaccines, that will be part of the exam 5, but this chapter 14 is, for me, the soul of this class, okay? So, very important. Do you have questions for me, issues with Connect, uh, or it's something that you are dealing with in the previous chapters? No? All good? Okay. So remember that this weekend we have exam 4, chapters 12, 13, and 14, right? So here we have viruses, we have the chapter 13 was related to protists, different eukaryotes that give you different diseases. We have vectors there too, and 50 questions as usual, including this chapter that is about, infectious diseases and epidemiology. This exam will be open during the weekend, okay? If you have issues, let me know, email me. Next week, there is no virtual session because it's holiday, so no activity. So pretty much the next session will be in 2 weeks, okay? Okay, so infectious diseases. I've been using this slide for years. And, I know that it's like a grim outlook, but it's very easy to forget that infectious diseases has been out there killing humans, other organisms too, right? And as we progress in the knowledge about how to stop them, how to create therapies, how to make vaccines, we are being able to reduce mortality, right? From 100 years ago, that was, one big one, the Spanish flu, that remember got that name because Spain was the only country reporting accurate numbers. That was the times of the World War One. So nobody was reporting accurate numbers because they thought that will take the morale down. And because the numbers looks high in Spain, this is what was named the Spanish flu. 50 million around the world. But after that, we start doing better, preventing the contagion, right? And today we are living the COVID-19, right? 5.2 million deaths around the world, and it's still counting. So we hope that after now having vaccines and also having the pill, we are going to be able to stop this faster, okay? But how is that infectious disease are a threat? Because they spread very fast. Now we live in more dense population, right? Most of the people today lives in the cities, and it's easier to travel long distances, right? And we will see that climate change is important, especially for the diseases transmitted by vectors, like insects, right? But now they can use wider habitats, right? They can move to different areas and transmit diseases as they spread in different areas, okay? So the first thing is to recognize that not all microbes interactions give us a disease, okay? So the way how this is understood from biological terms is symbiosis. That just means living together, not necessarily bad or good, right? So it's just living together. But there is four different kinds, okay? We have mutualism, commensalism, amensalism, and parasitism. So here, if you like the emojis, here you can have, these emoji thing that can help you to remember how, might be a benefit for both or benefit unaffected or benefit and harm. So let's take a look. The first one is mutualism. Here, both benefit. And I know it's kind of hard to think in, hey, how is that having microbes on my body? I have a benefit from that. Well, it's very much required if you want to absorb vitamin K, right? That is very much required for coagulation because thanks to E. coli, we are able to absorb vitamin K. Similar thing happened with vitamin B. We require the help of microorganisms that help us to absorb this that otherwise is not possible to take, okay? And of course, the microbe benefits because we provide an environment so they can grow from there. Commensalism is the kind when, for example, the host benefits or is not harmed, and the microbes, similar thing, benefits or is not harmed. And this, there is a lot of interaction that we are discovering with having a microbiota, that just having a diverse group of bacteria help us to, aggregate the bacteria that otherwise might just try to get through the barriers. Or we have this microbiota that creates surfactants, some kind of soap that prevents addition of the pathogenic bacteria, or by outcompeting this because creates, remember, substances that we can also use as antibiotics that help to, eliminate these bad bacteria or by direct competition. This is very clear in recent years. If you have, for example, your gut with a diverse group of bacteria that usually take care of the oxygen present in your small, large intestine, just because of that, is very, very hard for pathogenic bacteria to establish in your gut and create inflammation, okay? So normal microbiota, we have two major groups. We have resident microbiota, that what is considered normal. Pretty much we have it through the life. We will see next how this is acquired, or it could be transient, right? That remains for a short period of time, or is very related to, for example, I'm also going to show you examples of this, bacteria that is normal to have them on the skin, but if they cross the barrier of the skin, now is pathogenic, okay? So how do we acquire this microbiota? Usually, for example, during the birth process, we acquire many of them. And, this is very easy to establish, when you compare the microbiota of kids being born by C-section and normal delivery. You see that the bacterial diversity is quite different, right? And that happened because in different areas of our bodies, we have different microbiotas related to the skin, microbiota specific to the gut, microbiota specific to the genitalia. And during that third process, we acquire that microbiota, okay? It's so important that even in sub-Saharan Africa has been related to prevention, okay? Or actually given a higher chance to acquire HIV, right? To be infected by HIV if you have, a different kind of bacteria in the vaginal mucosa. So how is that some of these normal microbiota might become a pathogen? There is some factors related to the immune system that we are going to review in the next chapter. So, for example, having an injury, right? The kind of situation when the barrier is breached. So now that commensal is able to penetrate the barrier, right? And then you have an infection, like what happened with the Staphylococcus epidermolysis. We use this in the microbiology lab all the time to show you, how to grow, how to culture these bacteria. And usually if you are, a well-nourished person, your immune system is fine. This is BSL-1 microbe. But if you have malnutrition, if you are pregnant, or if you are taking medicines that reduce the numbers of your immune cells, now this is called immunosuppression because your immune system, right, the cells that watch your tissues, are in less number, or they are unable to act in the same way as they are normally do. And in that way, you are more exposed to have a disease by a pathogen or, by a microbe that usually is not creating a disease. Also, stress has been related to depression, right? Or, for example, what is more common today, by dysbiosis. This is when you take antibiotics that kills, right? Kills the normal microbiota, and now that diversity is gone, and you make room for other bacteria to establish in your gut, for example, and give you disease. So, for example, what you are seeing here, it's a large group of bacteria, and how that major groups are changed after taking antibiotics. This is in day 0, and this is after a couple of days, after a week, and you see that pretty much bacteroidetes and actinobacteria is gone, right? And after just a year, they come back. But when they come back, comes in uh, different numbers, you see? So this is very common, for example, after having a root canal treatment, and the doctor give you clindamycin, just to prevent you having an infection. But in your gut, there is big changes in the good microbiota, and that make you susceptible to severe infection, for example. Okay. The third kind of symbiotic relationship is parasitism. So this is the situation when the host is sick or even can die, and the microbe benefits. And the example that I gave you here is Helicobacter pylori, because we have no benefit at all of having Helicobacter in our bodies, but Helicobacter gets the benefits because they are able to use a system that is very similar to a needle to penetrate the cells and take iron, okay? So this is why Helicobacter pylori turns so aggressive. And if you have it, might give you a different spectrum of diseases. Might go from gastritis, an atrophy means that you have a outgrowth of epithelial cells, or give you ulcers, right? Now you have a perforation of the whole tissue, or Helicobacter pylori is the only biological agent that is related with cancer. Okay, this is so important that if you have Helicobacter pylori and you have, a different kind of cancer, not gastric cancer, lung cancer, skin cancer, this bacteria modulates so much your immune system that if you are being treated for Helicobacter at the same time that you receive the chemotherapy, the time that you require of chemotherapy can be cut to an half, because having this bacteria in your system modulates your immune system, right? Depress it, and then you are less able to fight cancer. You know, um, so it's pretty bad if you have it. Okay. Questions? No. Okay. So here another section of this chapter: reservoirs. So reservoirs of infections is those animals or places that maintain a source of infection. Okay, so have this connection in mind. Reservoirs are the source of infection. Similar, like when you have a water tank at home, this is your source of water. Here are reservoirs of infection. Okay, so could be animal reservoir, so those are called zoonosis, or you have humans, right? In this case, are called anthropozoonosis, or they are non-living, like sapronosis, could be bodies of water, could be dirt, could be metal. Okay, so animal reservoir zoonosis. Okay, many diseases are transmitted by farm animals, by wild animals, because we are in direct contact with the animal or the waste, okay? Or by eating the animal, right? For example, pigs is usually an animal that is able to transmit a lot of stuff to humans, because pigs, is very omnivore, right? Eats everything. So very easy to get infected with a lot of stuff. Other animals are birds because they move, right? We have migration, and when they migrate, brings different viruses, different infectious agents with them. So in this case, if you are dealing with a disease that is a zoonosis, and usually you have a farm animal, for example, dogs can also be a reservoir for different infections. Today has been analyzed that dogs or deers can also be infected with SARS-CoV-2. You know, so this now is also become a zoonotic besides of an anthroponic disease. So in this case, sometimes eradicate the infection is hard because some farmers won't give you away, the animal that is keeping the cycle of transmission in the farm. Okay, sometimes this is hard. Okay, so other diseases besides of what I just mentioned, tapeworm, fasciola, hepatica is one of these. Those are helminths, are worms. Protozoan, malaria, or toxoplasma. Remember, toxo is related to cats, but also, cattle can be infected with toxoplasma. fungal like ringworm, very common, be transmitted by domestic animals. Bacterial or viruses, for example, rabies. Rabies is one that is well known that is transmitted, zoonotic in terms of transmitted by bats, skunks, foxes, and dogs, but also by cats. Okay, there is a report a couple of years ago of kids petting a cat, in a elementary school, and the cat bites several kids and transmitted rabies to a couple of kids. Okay, so it's possible also cats transmitting rabies. Okay, so also anthropognosis in this case is a different animal, it's the human, right? For example, if you remember some years ago in 2016, we have Zika, right? We have Zika virus that this started from South America. Actually, this came from Asia, but becomes a big deal in Brazil after the World Cup, of the soccer World Cup there. And from there, it started spreading to the whole South America, Central America, even reach Florida, right? And in Florida, we have the transmission of all these different cycles here. We have the mosquito transmission, right? From by the uh, when the mosquito fed on a human, right? But also we have the sexual transmission from human to human, and this is male to female, male to male, female to male, right? It's harder female to female. We are going to see that later on portals of entry, right? But other ways how this happened was during pregnancy. If you remember that we have this situation of babies having very small heads, right? Microcephalia, right? And also by blood transfusion. And this is not the only one, right? This is parenteral. We are going to talk about this later. And also non-living reservoirs like soil, water, food, right, can also be focus of infection, right? Especially water, when you have a lot of cases coming up, popping in a city, usually the first thing that is being under surveillance is the water supply, because water is the kind of things that usually we trust, right? That the water treatment plant is doing a good job, right? That they are chlorinating the water, that they are using ozone also to decontaminate the water. And if something fails, right, these can spread very fast. So the book says that public officials could, I will say it must, right, notify the community, right, to boil water. And this is the kind of alerts that we have on our cell phones, from some months to months, to say, hey, watch out, boil your water. We are working in these pipes. So be aware, that the water might not be totally good for human consumption, right? Okay, so questions? No? Okay, so how is that these infectious agents, right, are able to get into our bodies? Here are going to mention definitions, going to provide examples, but please be aware that these definitions are very important. It might be on the exam, okay? So contamination is just the presence of these infectious agents, most of them microbes, right? Here or on the body, right? Just being on the surface means that you are contaminated, right? Now, the ability of the microbe to create an infection is a different thing, right? But if I using some of the microbiology methods that you are using in your virtual labs, you are able to isolate, to culture, to grow that microbe, means that you are contaminated, not necessarily that you are infected, right? Because it might be just on the surface. Infection now means that that infectious agents was able to get through the barriers, like defenses, right? Like the immune defense, the immune system, or the barriers. Okay, we are going to see that next chapter. There is different things that establishes a barrier, having mucins, having an intact epithelium, okay, that makes hard for these agents to go, but still they have different mechanisms that we are going to see next, usually related to virulence that allow them to get through the barriers, okay? And once they penetrate, overcome the barrier, multiplies and establishes, okay? This is very important, establishes in the body. Right there is when you can say, this is an infection, okay? It's not just a simple contact. Now you are infected because the infectious agents establishes in your body, okay? So here, with this figure, you have an idea of the different portals of entry, right? There is 3 majors, and I add one, that the book doesn't include. And the skin is one, having an intact skin is a very efficacious barrier that prevents you to be infected with different things. Remember that in different parts of the skin and also mucous membranes, right? We have epithelium. These epithelium have different factors that fight at that point or keeps at bay all these different microbes that are always present in the body, right? In the ear, conjunctiva, nose, mouth, the gut. Remember that this is also an epithelium, it's a cavity inside of your body, right? Another important portal of entry is the placenta and the parenteral root. So let's take a look. The skin. So the skin, remember, you have the epithelial, right? You have the epidermis, you have the dermis. So usually this give you a good thick, right? Several cells that this infectious agent needs to overcome. So usually the issue is because you have a cut, right? You have something that breaches, and through that, these infectious agents are now able to penetrate in rich areas that are not supposed to be there, right? There is some, assumptions. For example, Trypanosoma cruzi. This is a protozoa. Remember that chapter is a protozoa. Is transmitted by blood-sucking insects, and this trypanosoma is able to actually penetrate your skin. Okay. Do you think that this is something not seen in Texas? No, you are wrong. In the South, we have cases of Trypanosoma cruzi, and it's especially seen in dogs. Okay, so in this case, you have infectious agent that creates a bad thing in humans, and dogs could be a very important, reservoir. So it's also considered a zoonotic disease. Okay. Mucous membranes, remember, lines body cavities. So you have these membranes in your nasal cavity, in your mouth, along of your intestines, also in your lungs. All that have mucous membranes, right? And on top of these mucous membranes, you have mucus. That mucus is usually made of different things like antimicrobial peptides and other chemicals that kills bacteria, and also is thick enough to prevent the penetration of bacteria, right, to be in contact with the epithelial cells. Also, for example, in the stomach, remember our first chapters, just because the pH of the stomach is very acidic, that helps us to eliminate a bunch of microbes right there, okay? The additional road is the parenteral road. The book says that it's not a true portal of entry, right? Because means that the other portals were circumvented, but I will fight that, okay? Because today, if you, for example, share a needle, right? Like with drug addicts, with the opioids, when they share needles, a lot of these infectious agents can go right there, parenteral route into the bloodstream, right? Overcoming all these different barriers, right? So this is another way to penetrate, to overcome barriers. Placenta. This is what considered vertical transmission. So usually, for example, human to human, in the environment, when you sneeze and that virus goes into the air, right? Saliva droplets in the air, and other humans inhale those droplets with the virus. This is called horizontal transmission. But when you have placenta transmission, this is called vertical because it goes from parents to the descendants, right? And there is several infectious agents that can do that. Toxoplasma is a well-known, right? Bacteria like Treponema pallidum, syphilis, or listeriosis, right? Viruses, right? In RNA, DNA, RNA viruses, all this, is the kind of thing that can go through. Also, remember about Zika virus can also go through the placenta. Usually the placenta have their own immune system, okay, that watch and keeps at bay infectious agents that the mother may have and prevents the crossing of these infectious agents to the baby. Because the fetus, the baby, is just in construction. So the immune system is not even already there, right? So it's very susceptible if you have a pathogen that is able to breach, right? And of course, there is a lot of consequences for the fetus, for the baby, if the infectious agent is able to cross it. But what is these things that makes the infectious agents able to create the infection? So different factors, okay? This modification of these factors is what is known, like virulence, okay? That increase or reduce virulence. For example, for viruses are the ligands, okay? So those are proteins that establishes a relation with the host cells. So for example, in SARS-CoV-2, that is the virus, remember, that give you COVID, the SARS-CoV-2 have the spike protein that makes a connection, right? Creates an addition to ACE2, angiotensin receptor, and using that connection penetrates in the cell. In bacteria, it's different because in bacteria, we have a specific proteins, right, that also interacts and is able to, create infection. In protozoa, for example, here, you have this feeding disc, right, that damage the epithelial cells. What you are seeing here it's the small intestine. This is epithelial cells, and these feeding disc destroy the epithelial cells, and this, you know, is now able to make disease. Or what you are seeing here that looks like a horror movie, these are hooks and suckers from helmets, and they use these hooks to anchor to the small intestine, large intestine, and then feed, right? Just using, the nutrients that runs along of your gut. But these hooks is also a way to create adhesion to your tissues. Um, for bacteria, these factors are also very important to create biofilms. Okay, so usually here in these biofilms, you have some bacteria that are able to uh, bound first to the tissue. For example, here, this is the biofilm that makes cavities. So you have some of them that are able to first interact with the minerals in the tooth. But once you have this, like the first colonizers, you have on top of them growing, other bacteria that are called late colonizers. And from there, you have a permanent, you know, um, uh, group of bacteria growing into your uh, tissues. Okay. Questions?
No.
Yeah, all good. Okay. So now, more definitions. Okay. Nature of infectious disease. So remember that infection is created only after the infectious disease, right? This microbe invade, penetrates, overcome the body defenses and barriers, right? And now establishes, right? Establishes and growth. And then you may have or not a disease, right? So when you develop a disease means that some of your tissues, right, where this microbe is living, is using, now is being altered, right? Now they are not functionally as they used to. Now you have a abnormal function, right? And this is what is considered to be morbid. Morbid is you being counted as being sick, right? As having the disease, right? Because might be the chance, right, that you are showing nothing, right? So this brings, different ways to see disease. So you have symptoms, right, that are subjective. For example, um, you feel pain, but it's hard to, how do I measure pain? It's something that just the patient, right, can feel. So it's very subjective because you might say, well, that pain for me is 2 from 1 to 10, but for another patient is 10. That depends of, the um, different ways to perceive pain and experience. So those are symptoms, okay? Subjective characteristics. But science is very different because science is what we can measure, right? For example, I can measure fever, right? Use a thermometer, and I can measure your body temperature. And if it's higher than, right, 100.4, now you are in fever, right? Or I consider you suspicious if your body temperature is around 99, right? So you see, I can use this as an indicator, right? So those are called signs, right? For example, others are having a rash, red eyes, right? And you see that some of this, for example, when talking about the uh, 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 dengue, chikungunya, or Zika, all three having a viral origin. All of them are viruses, means transmitted by vectors. Some of these are being shared. Having fever is not significant because all these three give you fever. Having pain is also shared, so it's not enough to differentiate. What makes different? For example, in Zika, you have a rash, right? With chikungunya, now you have a severe joint pain, right? With dengue, now you may have nausea or vomiting, or specific red spots that is a little bit different from the skin rash of Zika. Okay, so remember, you might be infected, but could be asymptomatic, right? You are not being sick, you are disease free, right? Or you could be subclinical, means that for now, right? For now, you don't have the symptoms or all the symptoms. So it's a matter to wait, right? And this is what we are seeing today with COVID-19. That some people start with I feel fine, I feel normal, and then you have your smell is gone, right? You're no longer sensing things. And then it's like the first alert. Oh, I've stopped smelling things. So I better get tested, right? Because might indicate that you are getting COVID-19, right? So a syndrome now is the collection, right, of symptoms and signs that characterize a specific disease like what I just mentioned here, okay? Remember that etiology is related to the cause of disease. This is related to the Koch postulates that we review back there in the first chapters. And this is what is so important to be diagnosed, right? To rule out. For example, you may have fever, right? You might have some joint pain, and you might say, oh, I may have COVID. So you better go test it, right? And then they say, you are SARS-CoV-2 negative. Oh, okay. So might be that you are not fully developing, yet the whole symptoms. You are just in the initial stage, not enough virus in the sampling area to be detected, or you are being attacked by a different virus, right? And this is why today they are sampling or testing for the two, for SARS-CoV-2 and also for different flu viruses, right? In order to tell you what you are feeling is COVID-19. What you are having right now is the flu, right? And according to that, you may have a different treatment, right? So how is that these infectious agents give you this differential disease, right? This is what is called virulence. Virulence might relate to pathogenicity, means how much sick are you, right? Because, for example, for C. diff, right, that is virulent. According to the strain, you may be infected with a strain that makes no toxins, so this is fine, or make just one of the two toxins, right? And then you have a mild disease like the UK 630 strain, or you are being infected by NAP1. And this is the strain that makes two or three different toxins and give you severe disease, right? Now you have a full-blown pathogenicity in your tissues. So these virulence, might be addition factors, might be related to higher rate of infectivity, like what we are seeing today with SARS-CoV-2, because some of these Delta variant, in the case of SARS-CoV-2, are able to transmit faster, right? Because they have proteases that are able to release the virus faster from the host cell, or because they live in communities like biofilms, making extracellular enzymes that we are going to see. They are able to make toxins or not of what I just explained with CDF, or things that prevents, to be taken by the immune system. So about the enzymes, there is different bacteria that use, for example, the hyaluronidase. This is an enzyme that is used by the bacteria to bridge through the epithelium and then gets into your tissues or coagulates. In this case, your body detects the bacteria and creates an area that contains the bacteria, okay? Usually with a bunch of fibers around. So that creates like a cloth, okay? And the bacteria, using coagulases and kinases, are able to destroy these fibers and being released from that cloth and then spread in the host, or because make toxins. So CDF, what I just explained, makes exotoxins. So the kind of toxins that are released in the environment by the bacteria, but these toxins kills the cells. And once the cell is gone, now you have a place, an empty place that can be used by the bacteria to bridge and gain access to your tissues, right? Other bacteria export these toxins. So now the toxin is not being staying there, it's not in the tissue, it's going through, for example, here, the bloodstream, right? And give you pathogenesis, my damaged cells, for example, in the liver is very common, okay? Here, other examples of exotoxins, like for example, for botulism, that the toxin blocks the release of acetylcholine. Remember that acetylcholine is a neurotransmitter. So usually you see these droopy eyes, right? Because it's hard for the muscles to contract. Or the opposite, like tetanus. In this case, the toxin, right, prevents the release of GABA. So now the muscles are in a permanent contraction, okay? Other factors that are considered virulent, remember in the case of tuberculosis, you have this acidic component of the capsule or at the wall of the bacteria that makes these bacteria slippery. So it's hard for the immune system to trap them and phagocytize them and destroy them. Or because they have factors, right, that allow them to survive the attack of the lysosomes, all these lytic, dangerous components of the lysosome, acidic, hydrogen peroxide, enzymes being released from the lysosome, and the bacteria survive and from there reproduce and destroy the cells, right? Questions? No? Okay. So the stages of infectious diseases, usually during the incubation period, once you get infected, right? Once the infectious agent overcome the barriers and start multiplying, initially, the first period, right, is in so low numbers that it's called incubation, right? You are not being detected even for, if you are being sampled, you might turn negative because, the virus are in low numbers, as you can see here, right? For some infectious agents, right, for example, for a bacteria, could be less than a day. Some viruses could be from, a couple of days, usually 5 days. For COVID-19, could be, something from 4 days to a week. And then we have illness. And here is where most of the signs and symptoms appear, right? This is what is called illness, because now you see the effects of the growing, right? Exponential growing of these infectious agents in your body, right? Now they start affecting the functioning of the tissues, creating damage on the tissues, right? And this is what makes you feel ill, right? Sick. And then here you have the turning point. What is this turning point? When your immune system is now being able to learn and fully deploy an attack using um, um, antibodies, using T cells that are able to destroy that pathogen. Usually that takes between 1 to 2 weeks, okay? This is why the quarantine period of the COVID-19 is 2 weeks, because this is the time that your adaptive immune system takes to learn from that microbe or the virus in this case, and makes the antibodies that are able to neutralize, to prevent the attachment of the virus to the host cells, okay? So this is why usually this period is around 2 weeks, okay? And then now that your immune system is fully functional, or you are taking antibiotics, now you see the decline, right? That is related with the decline in the microbe, right? In less microbes, less viruses, less bacteria. And at the end, convalescence. This is when you start recovering. Now, here's when you say, hey, I feel better. Not totally normal, but I feel much, much better, right? Okay. So now, how is that these pathogens can go out of the body? This is what is called portals of exit, right? And here we have different ways how they can go out, right? So by the flakes of the skin, right? Remember that we have a bunch of bacteria living in the skin. So in the flakes, there you go. You have bacteria also going there, right? By broken skin, now you have some microbes living in the blood. And now that that blood is, leaving your body, now could be in direct contact with another person that can get the infection from there, okay? And this is, related with, for example, HIV, right? Usually during the HIV, that is sexual transmitted disease, you have that as long as the epithelium, right? And both ends in this sexual relation are being intact, you shouldn't have any infections, right? But during the sexual intercourse, it's very common to have friction, right? And that friction now is broken epithelium, and you might bleed, okay? You might not see it, but you might be bleeding. And now having that interchange of blood is what allowed the transmission of these viral diseases, okay? And this is usually easier when it's through the anus, okay? Because if you compare, if you remember your MP classes, in the anus, you have a lower number of cells, right? Making that barrier compared to the vaginal area, okay? There is a thicker barrier in the vaginal area compared to the anus. This is why some viruses are able to penetrate much, much easier through the anus compared to the vaginal area, okay? Earwax, I know, sounds weird, but for example, earwax is one place that Helicobacter pylori has been found, okay? Tears, nose secretions, mouth, saliva, very common for viruses. Mammary glands, milk secretions, yes, through the milk is also possible. Urethra, seminal vesicles, semen lubricating secretions. Some of these are able to go right with the semen, others just through the blood, okay? Modes of transmission. So if we have person to person, this is direct. So we have skin to skin, congenital that we review through the placenta, or per act, like sexual transmitted diseases, right? Could be also indirect when it's by droplets, right? Like what we are seeing today, right? SARS-CoV-2 being in the droplets of saliva in the environment, and this is what we are using mask, right? To prevent these droplets in the air, right? In order to contain these droplets inside of the mask. Or fomites. This is very common on healthcare settings. Remember CDF, you are going to hear this in hospitals all the time because fomites is that kind of element, a doorknob, a wall that is able to hold the spores of bacteria. And once someone else touches and put that finger or hand in their mouth, boom, you got the infection, okay? Or by vehicle means, for example, waterborne like Giardia or cholera, foodborne, salmonella, vector-borne, remember, because you have an insect, mechanical tetanus. This mechanical, remember, is when you have a nail, you have spores of this bacteria present in the rusty metal. So if you step on that, now your skin is being breached. Airborne, right, like anthrax that you have. This is not common, but if it's weaponized, now you can use anthrax in particles in the air. Usually, it's so big that it's not transmitted in the air, but this is the risk with anthrax that can be weaponized, okay? Questions?
No, no questions.
Okay, so classification of infectious diseases. There is different ways to classify diseases, okay? So by duration of severity could be acute, right? Means that these develop in a couple of days and then you shake it also after some days. So usually less than 2 weeks is considered acute. Chronic usually means months, okay? It's not measured in weeks, usually in months, several months. And now this, that is kind of weird, subacute. Subacute means something in the middle between acute and chronic. So something that lasts for less than a month, okay? Something between a couple of weeks and a month. latent, like herpes. This is the kind of thing that you be infected for life and it stays in your nervous system. And when your immune system goes down, right, from the nervous system, now this expands and now you have the disease. And then immune system goes up, control this. Now the virus stays again into the nervous system and it stays there for years, right? This is latent. Or degenerative, if it's the kind of things that stayed with you through the lifetime, it might compromise the systems or tissues onto the point that, is fully compromised. By pathogen, if it's viral, bacterial, parasite, usually like a protozoa, like for example, toxoplasma or trypanosoma, fungal, by fungi, right? By the system, if it's nutritional, mental, or immunological, if in this case the immune system is depressed, right? Or the autoimmune diseases. Now is your immune system being very active and is damaging, right? Is damaging the tissue. And here, a category that is very important, okay? Communicable means that it's infectious, okay? And this is mandatory to be reported to the local, state, and federal authorities, right? Because they go by modes of transmission. If it's congenital, remember, or nosocomial. What is nosocomial? Is the kind of disease that is transmitted in healthcare settings. Okay, that means the hospital, a healthcare center, senior living facility, right? This is nosocomial. This is different. Iatrogenic is the kind of disease, in this case, not always caused by an infectious agent, but the iatrogenic is the kind of thing that happens when you have a medical procedure and then you end up with a different pathology. Okay, so for example, you have malpractice, the kind of thing that the doctors did correctly, and you end up having another illness because of the procedure. Okay, or it could be primary or secondary infection, according to how these infectious agents, goes in line. It's very common for some patients in hospitals to be hospitalized because you have a viral infection and then you have an additional infection acquired in the hospital, right? Okay, if it's not non-communicable, like a tooth decay of some kind of something that you inherit from your parents, this is non-communicable. Okay, epidemiology. So you are going to be part of this, collecting data, that is very important for epidemiological studies. And here, some definitions that are very important to understand. How is that epidemiology works, or what they are calculating. So in epidemiology, they have this analogy. The epidemiologist bathtub, okay? Incidence is just the number of new cases and is usually related to a specific area or population. So for example, incidence of COVID cases on Galveston area, Galveston County, or Texas. You see, it's just specific for the population of a city or for a territory, okay? Now, Prevalence is different because in prevalence, you have a cumulative number of cases, right? Because every day we have new people being infected with COVID, right? But the prevalence is this total number of people that are, just feeling sick, people that are fighting the disease at home or at the hospitals, or the people that are getting better but still have it. So this is prevalence. This is total number of cases, and it's in the same way area of population. So these, remember, patient here, so could recover or they might die, right? And this is when we can also calculate this, the recovery rate or the rate of mortality. So from this prevalence that give us the total number of cases, we can calculate the rate of the percentage of patients that are dying after getting the disease, or the percentage of patients recovering after having the disease, right? And occurrence is related to the geographic distribution. I will show you this in a couple of slides from here. So here, a graphical way for you guys to understand the difference between incidence and prevalence, okay? This is HIV AIDS cases on US among adults. Incidence, as you can see here, spike, right? There were a high transmission in early 90s, right? And but through the time, the prevalence, right? Because this is a chronic disease, is very easy to calculate, who's getting the sickness, right? And how this been accumulating over the time. You see that the prevalence increases. Doesn't matter that the incidence is going down, you still have new cases coming every month. And this is why the prevalence increases, right? Until the point that now you have different control measures, right? In the case of HIV, there is no vaccines, but we have different treatments, right? We have chemicals that prevents the virus to keep infecting cells. And this is the turning point when you see that prevalence start going down. Otherwise, prevalence just go down because the mortality might be too high, right? Like, for example, with Ebola. Epidemiologists usually show you this in different ways, like, for example, a heat map that give you like total cases by area, by state, right? Of the percentage of cases through the time, or a number of deaths according to the age groups, right? And here the occurrence, right? How is that these diseases can spread in terms of, an area? And these can be used, here I'm showing you the whole continent, the Americas, but can be also, just in a smaller territory. So you have an endemic disease when you have this happening through time, right? For example, we have the flu, right? Flu is endemic. This happens across the Americas and is kind of permanent, right? During the whole year, we have this, usually seasonal, right? Usually is related to winter. But for example, this year, we have the peak moving late, right? Move to spring. Could be sporadic, right? If this is the kind of things that sometimes jumps, right? For example, we have last chapter I was mentioning um, West Nile. Okay, West Nile it's sporadic. We know that in some areas, might be present year long, but not always making big numbers. But according to the, um, if they are using, um, if they are using um, pesticides, right? If they are using, you know, uh, if they are taking control measures for vectors, right? Using insecticides, um, uh, that takes the numbers down of these insects. You may have a better control of this or not. Epidemic, usually, like for example, what happened with COVID-19, this started in China, right? So was very concentrated in an area, but from there later turned into a pandemic, right? Because it spread through the whole continent. So this is something that, for example, not only been seen with COVID-19, this also happened in the previous years with Zika that started here in Brazil and later spanned through the whole continent. These also happened in the past with malaria, yellow fever, because using pesticides, we were able to control the vector as objective in the whole continent. But because one country decided not to fumigate, right, not to use this, from there, we have again the disease and the vectors spreading through the whole continent. Epidemiology studies, we go from descriptive epidemiology, that is just locating the cases, right? Please take a look on the video that I post in Canvas about John Snow. He was the first epidemiologist. Um, Actually, I will have to say that epidemiology became a thing here on the United States more than 100 years ago. Was this country the one that pushed epidemiology, on the spot? But they were looking for a hero. So they looked for someone to, exemplify that kind of work. So John Snow, in the 1854, pretty much did a very good scientific work by looking the source of cholera in London. So please take a look. It's a very nice video about how descriptive epidemiology can be used to trace, the source, uh, what is creating that disease, where is the patients being showing how is the distribution of these patients, okay? So all these brown dots that you are seeing here, it's the patients, and he was suspicious about the cholera, which is a bacterial disease, was spread through the water pumps, okay? Because most of the cases happening around to this pump, okay? So please take a look. It's a very nice video, okay? But epidemiology has matured, has discipline. So now we have analytical epidemiology that goes beyond, right? Looks for the probable cause, modes of transmission, methods of prevention, right? Could be retrospective, right? How these happened before, right? Or experimental epidemiology. Now they can have the chance to test, right? How this happened, in different countries, what they are doing here that is not done in a different country, and how that can be associated with having a better outcome or to a poor outcome, right? We are seeing this today in action, right? Some countries in Europe decide not to do lockdown. And at the beginning, they say, well, now we are doing fine. And then, bloom, you have this huge wave of cases, and they did worse, right? So we are seeing this in motion today. And of course, hospital epidemiology, nosocomial studies, you are going to be a big part of this, collecting evidence, collecting data, right? About if these infections that you are having in the hospital have exogenous, right? Comes from the outside of the hospital environment, or if it's something endogenous. This is why today has been said that hospitals, facilities have like their own microbiota. If it's related to be iatrogenic, related to medical procedures, like for example, a doctor that tell you, hey, take this antibiotic, for your root canal and prevent an infection that give you, a disease like C. diff, or by a surgery. And what uh, measures are you going to put in place, right? And remember, most of the time is you guys, the one that is going to be in charge of executing these measures, okay? And the point that you don't want to go, super infections, right? When now you have these microbes that are resistant to a lot of different antibiotics, right? And they are able to go beyond of these control measures. Okay. Usually, to control them requires aggressive. Remember what I told you? Just hand washing is good controlling several diseases, right? The transmission of several pathogens. But if they are not put in place or being watched, right? Police, many times I told you, I saw a hospital that have this out of control, and they have to put a nurse right there at the spot to watch that everyone, including the most renowned doctors, yes, they have to wash their hands, right? They have to put someone to see that if they are doing the correct hand washing, okay? And of course, this data must be shared, right? In order to alert not only our hospitals around you, but also the city, the state, in order to put this under control, okay? There is public agencies that have an active role in this. Here in Texas, we have several at the county level. We have a very important one that usually provide guidance, right? What kind of things many facilities can do or not, right? But also in Canvas, I post a very nice video about how CDC responds to outbreaks, not only on the States, also outside of the States. And this, is a very important agency that brings these diseases under control. And of course, public health education. This is another part of your job. You are going to be part of this to bring education, right? To design strategies to prevent the transmission by education. Okay. And this is it. Do you have questions?
No.
No? Okay. So now I'm going to do practice questions. Okay. Practice questions. So remember that you can put in the chat box your answer. Okay. So it's a symbiotic relationship in which one organism benefits and the other organism neither benefits or is harmed. So how do we call that?
Commensalism.
Commensalism. Very good. Which of the following is an example of mutualism?
Bacteria living in a human colon.
Very good. Bacteria living in the human colon, right? Because in the other ones, usually is related to a disease, right? Microorganisms that are able to colonize the skin or mucosal surfaces for a limited period of time are called, the key word here is this one, limited.
Transient microbiota.
Very good. Transient, right? Transient. Okay. The term that means free of contaminants. Um, here, oh, okay. This might be more related to a different question, but no, it's the publisher who made these questions, not me.
I
So
want
what?
to say arsenic because whenever I hear arsenic, I think arsenic.
Yes. Yep. Axanic. So means free of contaminants. Okay. Usually means the kind of surface, the kind of environment that is free of living things. Okay. This is another way to see, the presence or absence of microorganisms. Okay, which of the following are conditions in the host that cause normal microbiota to become opportunistic pathogens?
All of the above. I didn't want to keep answering the same questions.
Very good. All of the above. So how is that hormonal changes can take this, give a chance to opportunistic pathogens? Well, think on pregnancy, okay? Because during pregnancy, hormones reduce the ability of the immune system to fight infections in order to do not attack the baby. Remember that half of the genetic material of the baby do not belong to the mother, right? Is from a different person. So this is why it's so important that the hormones, you know, tune down the immune system to protect, uh, you know, to avoid harm in the baby. Okay. What is a zoonotic disease? Zoonotic is from animals to humans. Okay. Doesn't matter if it's exotic, domesticated, farm, you know, um, they just have the disease. So, for example, if you are working with animals in a zoo, in a hospital, in a research facility, you might be asked, okay, doesn't matter how good is your immune system or not, you are going to be asked to take some vaccines because it's very common that many of these animals have are reservoirs, of these different infectious agents. For example, hepatitis B, very common in animals. Um, which of the following is an example of parenteral route of infection? parenteral. If it's parenteral, remember, requires to overcome, it's almost like a free pass to overcome all these barriers, right? So it's stepping on a nail, right? Because right there, the nail penetrates all the barriers and take this bacteria right there into the tissues. The most frequently used portal of entry is the respiratory tract, okay? Is the most frequent because there you have, a very frequent movement of air, right? Happen during a lifetime. So you have a high, um, um, exposure, and the epithelium have mucins, but according to your state, that mucin could be thick, could be thin. In your lungs, now you have that the alveoli is just one cell thick, right? And you have a bunch of capillaries. So you see the barriers there are not that thick, right? It's, kind of simple barriers to overcome. And also these, frequent exchange, right, of air in, out during the lifetime make this the most frequently portal of entry. Okay, the terms that applies when the multiplication of a pathogen results in a negative effect on the body is so antagonism for sure is not, right? Synergy, because synergy is when 1 + 1 = 3, right? So what is infection of disease? Is disease, right? Because you might be infected, but might be that you are able to neutralize, to prevent, right, the full development of the infectious agent in your body, right? You stop it at bay. So might infect you, but it's not creating a disease. You can get rid of that easily. What is the term used to describe characteristics of a disease that are felt only by the patient?
Symptoms.
Very good. Symptoms, right? Like pain, okay? This is the classical, okay? Only the patient can say how strong is the pain. Fetal alcohol syndrome is an example of what kind of disease? Congenital, right? Because it's the consumption of alcohol by the mother, right? That create changes in the baby, right? By the exposure. Why are bacterial capsules effective as virulent factors?
D.
Because you have that these capsules might be very slippery, right? Or make them high, right? For these microbes away from the immune system. Which of the following types of toxins are produced only by gram-negative bacteria? I have that in the slides, but I didn't mention it. Endotoxins, okay? Endotoxins, gram-negative. Which of the following is an example of portal of exit? All of them, right? Blood, urine, secretions, all these are exits. Which is true about mechanical vectors, like needles. Let's see, right, that they carry pathogens. Remember, mechanical vectors, because they are a reservoir, right, of these pathogens there. Is not alive, but still have them. All of the following are examples of direct contact transmission of disease, except so this is easy, right? Because he's sharing a drinking glass. Because now the glass is the vector, right? It's not direct, it's indirect. The common cold is an example of what kind of disease? Remember.
Acute.
If it's less than 2 weeks, between
Is it
2
acute,
weeks,
Professor?
it's acute, right? Because usually the common cold, you shake it after a week, right? So it's acute. Acute, usually less than 2 weeks. Subacute, something between 2 weeks and a month. Chronic, months, right? What type of disease develops rapidly, but only lasts for a short time? Acute, right? Short time, acute. What term best describes the total number of disease cases in an area during a given period of time? Total number?
Prevalence.
Very good. Prevalence. What term best describes a disease that normally occurs continually and a relatively stable incidence? Continually. It's endemic, right? Endemic. Which of the following best describes an endogenous nosocomial infection? Nosocomial means, right, the microbiota becoming opportunistic in the healthcare setting, okay? Healthcare setting. This is what describes, you need to associate with nosocomial happening at the healthcare setting. Whose responsibility is to control disease transmission and spread? Everyone, right? The individual, public health officials, and the agencies. Okay, this is it. So do you have questions? No? All good?
Yes, sir. Thank you,
Okay,
Professor.
so you're very welcome. Remember that exam will open this weekend, okay?
Okay, have a happy Thanksgiving.
Thank you. You too, guys. Bye-bye.
Thank you. Bye-bye.